Good morning everybody. Today we will discuss about properties of amino acid. It is the properties of amino acid that determines the properties of protein. Amino acids are colorless, crystalline, soluble in water, but not in organic solvents. They may be sweet like glycine, alanine, valine, or tasteless like leucine. They may be bitter like arginine and isoleucine. You have heard of, might have heard about the monosodium glutamate, Ajinomoto. Ajinomoto is monosodium glutamate. It is very sweet and not sweet, it is a taste maker rather. It is used in the Chinese restaurant to make noodles more tasty. But sometimes this taste maker, monosodium glutamate, it is not tolerated by some people. They may produce short time feverish like syndrome, that is called Chinese restaurant syndrome. During our last class, we have seen that those amino acids having chiral carbons, they exhibit optical property. Except glycine, all the amino acids, they exhibit optical property. They behave like ampholytes. Ampholytes are the molecule have both acidic and basic groups. And this type of molecule, they act as buffer. And amino acids also in protein, they act as buffer. In case of acid-base regulation of blood, they plays a role in buffering the change of pH. Deuterium or dipolar ion. Amino acid, we have seen, they have positive ion in amino group and negative ion in carboxyl group. When we go on adding acid, they can accept protons. So to some extent, there will not be any change of pH of a protein solution or solution containing amino acid. So also, when we add alkali, they can donate proton and there will not be any change of pH. In a particular pH, amino acid exhibit equal number of positive ion and negative ion, where net charge of amino acid remains 0. That pH is called isoelectric pH. That's why net charge of amino acid depends on pH medium. Strong acid amino acids show positively charged, and strong alkaline pH, amino acids show negatively charged. Here, this is an amino acid. When acid is added, it can accept proton, and when base is added, it can donate proton. So, this type of molecule is called deuterium. An amino acid exhibits this property, so amino acid also is called a deuterium. Chemical general reactions of amino acids are due to functional groups of amino acids, carboxyl group and amino group. They can produce bonds with the nearby amino acid. Here one amino acid is there, here another is there, so they can produce a bond here that is called peptide bond, and the residue amino acids are called residue amino acids. Now, reactions due to carboxyl group, its carboxyl group can be removed and forms the corresponding amines like histamine, tyramine, gamma amino, butyric acid. Reactions with amino group, reactions with ammonia such as aspartic acid, when reacts with ammonia, it gives asparagine. Due to amino group, transamination reaction, oxidative deamination and formation of carbamino compound. The amino group of amino acid, this amino group of amino acid can be shifted to a keto acid and a new amino acid is formed. Keto acid converted into amino acid, and amino acid which took part in the reaction become again a new keto acid. Here alanine is an amino acid, pyruvate, this is a keto acid. In presence of coenzyme PLP, pyridoxal phosphate, it is the active form of vitamin B6. Vitamin B6, its coenzyme is pyridoxal phosphate. It is very important in amino acid or protein metabolism. So, in presence of this coenzyme, the amino group of alanine is transferred to pyruvate, forming a new amino acid, glutamate, and the alanine, it become a new keto acid, alpha ketoglutarate. This is alpha ketoglutarate. Here you will get short note on this decarboxylation of amino acid, then transamination, oxidative deamination, you may get short note. In oxidative deamination, the amino group is removed to form keto acid and ammonia. In first one, there is a transamination. From one amino acid to keto acid we transferred, and amino acid became keto acid and keto acid became amino acid. But in case of oxidative deamination, Here amino group is released as ammonia. So the amino acid, it becomes keto acid, product it keto acid and ammonia. Formation of carbamino compound, addition of carbon dioxide to the amino group forms carbamino compound. In case of hemoglobin, carbon dioxide carried by formation of carbamino compound. So now we will see some biologically important peptides. Here also you will get short notes, long questions from this topic. Peptides. We know what is protein. These are the polymers of amino acids bind together via peptide linkage. These are nothing but polypeptides. Then we have again mentioned here polypeptide. So what is the difference? Difference is that protein will contain amino acid in terms of thousands, but peptide will contain less than 1000. Less than 1000 polypeptide, when there is amino acid attached, these are called polypeptides. Then peptides, in case of peptide, the number of amino acid will be 10 or below. 10 or below. And in all cases, there will be peptide linkage. So, coming to the peptides, when there are two amino acids, it is called dipeptide, and there will be one peptide linkage between in the dipeptide. So in case of tripeptide, there will be 3 amino acid, 2 peptide linkage. Likewise, pentapeptide, hexapeptide, hepta, octa, nano. In nano, how many amino acids? 9. And peptide linkage 9 minus 1. Now, last is deca. Dipeptide, examples are carnosine, anserine. Another dipeptide is there. You have heard sugar free. Sugar free, you have heard of the word sugar free. The sweetening agent used by the diabetic patient in place of sugar. It is nothing but a dipeptide. It is 200 times more sweet than sugar. Tripeptide, example is glutathione, thyrotropin-releasing hormone. Glutathione is a tripeptide synthesized by glutamate, cysteine and glycine. Cysteine has disulfide bonds. So, glutathione, it may remain in two forms, oxidized form and reduced form. Now, glutathione, this molecule is very important for the organism, for the living body. This is abundantly present in the nature because every now and then the cell need glutathione for its survival. to avoid cell toxicity by the free radicals generated in the biochemical reactions of the cell. Now, this reduced glutathione, it is present in abundant number in relation to oxidized. Say, reduced glutathione, it is available 100 times more than the oxidized one, and an enzyme called glutathione reductase, it converts the oxidized glutathione into reduced form. Reversible oxidation reduction of glutathione is very much important for cell survival. Now, functions of glutathione, it serves as coenzyme. Example, prostaglandin PGE2 synthetase. During the process of synthesis of prostaglandin, it prevents oxidation of sulfidyl groups of several proteins to maintain protein function. For protein function, the required conformation is very much important. Now, due to production of free radical in the body, if the sulfidyl group or disulfide bond of the protein is oxidized, then the protein conformation will be lost, and that protein will not have any more functionally capable. To prevent this, it is the glutathione, it remains ready to accept the assault caused by the free radicals. So, as soon as there is free radical like hydrogen peroxide, the glutathione, it reacts with the hydrogen peroxide and become oxidized. And in next turn, this oxidized glutathione by the enzyme glutathione reductase, it again comes back to its reduced form. So, to maintain protein function, it plays a great role by preventing oxidation of sulfidyl groups, maintains RBC membrane integrity by keeping the ferrous ion as ferrous state. because ferric state is injurious for the RBC membrane. Protects hemoglobin from getting oxidized by an agent such as hydrogen peroxide. Transportation of amino acid in intestine and renal tubule via gamma-glutamyl cycle or Meister cycle, that you will know when transportation of amino acid through renal tubule is taught. So, it is the glutathione which transport amino acid through the cell membrane in intestine and renal tubule. Take part in detoxification such as organophosphates, nitro compound, converted in mercaptopuric acid, converted into mercaptopuric acid. See, when you take food, plenty of molecules which are injurious to health are taken along with food. From the intestine, whatever the food particle nutritionally active molecules are absorbed, it has to pass through liver, and liver is the site where detoxification is carried out. So, in case of detoxification, one of the molecule is very important. This is the glutathione. The metallic poisons, the organophosphorus compound, then nitro compound, when they pass, they are converted into mercaptopuric acid, so that they are liver cells are protected from their toxic effect. Toxic amount of peroxide, free radical produced in cell are scavenged by glutathione peroxidase. It is a selenium containing enzyme that you will learn when you are taught chapter of free radical metabolism. So, here the reaction happens like that. Two molecules of glutathione with hydrogen peroxide, they react, and it is accomplished by glutathione peroxidase enzyme, and glutathione is oxidized and water molecule is removed. And again in the next reaction, this glutathione, oxidized glutathione, it is converted into reduced glutathione by glutathione reductase. Now, next tripeptide is renin, angiotensin. Sorry, thyrotropin-releasing factor. Now, thyrotropin-releasing factor, it is secreted by the hypothalamus. It stimulates pituitary gland to release thyroid-releasing hormone, and that thyroid-releasing hormone acts on thyroid gland, and thyroid hormones are released. Now, this is a tripeptide released from the hypothalamus. This is important tripeptide. Now, pentapeptide, enkephalin. Enkephalin, see, in our body, continuous wearing and tearing is going on, and pain producing substances are produced, but we do not feel pain because of this enkephalin. It is naturally produced in the body. It is an analgesic. It is opioid analgesic type. That means it acts centrally. So, because of this enkephalin, we do not feel pain. We feel happy because of this enkephalin. Now, important peptide, octapeptide. This is angiotensin 2. Now angiotensin, when there is a volume depletion in the body, in extracellular fluid or in the intravascular fluid, the juxtaglomerular apparatus of the nephron, it gets activated or it gets sensitized and it releases renin. Now this renin, it converts this tensiogen into angiotensin. And angiotensin 1 is again by the enzyme angiotensin converting enzyme, it converted into angiotensin 2. And angiotensin 2, this causes release of aldosterone. And aldosterone, it retains sodium in the body, and along with sodium there is retention of water. So there is conservation of water in the body and volume of extracellular fluid or the intravascular fluid is replenished by this way. So the angiotensinogen is a 10 amino acid peptide. By renin, 2 amino acids are removed, it becomes angiotensin 1. Again by angiotensin converting enzyme, 2 peptides are removed, 2 amino acids are removed, it becomes angiotensin 2 and it is angiotensin 2 which is functional. Now in some patients, due to the hyperactivity of the aldosterone, sodium is retained, volume of extracellular fluid and intravascular fluid component increases, so there is high blood pressure. High blood pressure is there. In this case, what we do, we give antihypertensive to inhibit this angiotensin converting enzyme. So if angiotensin converting enzyme is inhibited, then formation of angiotensin 2 will be reduced, thereby reducing the retention of sodium and water. So there will be volume depletion, pressure will come down. Now such type of examples are captopril, enalapril, they are the angiotensin converting enzyme inhibitor. Vasopressin is a 9 amino acid peptide. It is also called antidiuretic hormone. Antidiuretic hormone released from the posterior pituitary. This is also secreted when there is a volume depletion in the body. So antidiuretic means it prevents excessive urination and retains more water in the body. So its function is retention of water. Another is oxytocin, 9 amino acid peptide. Its function is to cause uterus to contract. This become active when there is a labor pain. When there is a term pregnancy, the oxytocin is released and it causes uterine contraction. When there is inadequate oxytocin secretion, then from outside we give oxytocin in drip to increase the labor pain. This is called augmentation of labor. Another is bradykinin. This is a vasodilator. Now let's see the properties of protein. Properties of protein are nothing but the properties of amino acid collectively. These are usually tasteless, odorless, but on heating, it produces a burnt feather-like smell. Molecular weight, it is the summation of molecular weight of the amino acids. One amino acid weight about 110 kilodalton. So, a protein containing thousands of amino acid will have molecular weight 110 into number of amino acids. Insulin have molecular weight 5700 kilodalton, albumin has 69,000 kilodalton, like that. Protein are soluble in water and they form colloidal solutions in water. Viscosity, in solution they produce viscosity, and fibrous proteins are more viscous than the globular proteins. Amphoteric in nature, that means in solution they exhibit both positive charge and negative charge. Hydration, when in solution proteins they absorb water and produce a wet shell like layer around it. This is called hydration. Any physical or chemical agent which disturbs the hydration of the protein causes precipitation of that protein. Precipitation of protein. Factor that neutralizes charges or removes water shell will cause precipitation. Mineral acid denaturates protein, so it will cause precipitation. Ammonium sulfate, sodium sulfate removes water shell, and that is also will result in precipitation of the protein. Organic solvent, it dehydrates the protein, so it will cause precipitation. Now, protein, as it is amphoteric in nature, in a given pH, some protein will exhibit equal number of cations and anions, I mean positive charge and negative charge. So, in that pH is called isoelectric pH for that protein. Casein, it has an isoelectric pH of 4.6. So, in that pH, isoelectric pH, the net charge of the protein remains 0. Protein's solubility becomes minimum and it gets precipitated. And this is the technique used for formation of curd. The pH of milk is reduced due to production of lactic acid by the lactobacillus, and it brings the pH at around 4.6. So, the milk protein casein gets precipitated. That is the formula of, that is the principle of curd formation. TCA, tungstic acid, sulfosalicylic acid, heavy metals like mercury, lead precipitate proteins. When you go for your practical, when you do estimation of creatinine, we add with the serum tungstic acid that causes precipitation of the serum protein, and we go for the test after straining the solution and removing the protein part, because protein hampers in estimation of creatinine. So this principle of precipitating protein by the tungstic acid is used in case of creatinine estimation. By heat, it is a physical method that also causes precipitation of the protein. Significance of precipitation of protein: we detect whether solution contains protein or not. When you do your urine examination, you have seen, you have done heat coagulation test, is it? Heat coagulation test, have you done? Have you done heat coagulation test for during your urine test in practical? Yeah. So in that case, we upper part of the urine sample we allow to boil, and that causes precipitation of the protein due to coagulation. So we detect whether protein is present or not. To prepare protein-free filtrate, what I told, phosphotungstic acid we add in the serum and we precipitate the protein and remove the protein part. So protein-free filtrate we take for estimation of creatinine. Separation of albumin and globulin by full saturation and half saturation. When in a serum solution, in a solution adding serum, we add sodium chloride. At half saturation, there will be precipitation of some protein, that is globin. That globin will be precipitated in globulin will be precipitated in half saturation of sodium chloride solution, and albumin will be precipitated at full saturation. So by this way, we can separate albumin and globulin from the solution. To precipitate cations in the heavy metal poisoning, when somebody ingests accidentally heavy metal, so we allow them to take egg white. Egg white contains protein, and the heavy metals, they will oxidize the protein and get bind there. So it will not be able to absorb from the intestine in the system. So poisoning can be prevented. Color reactions of protein. Just what are the color reaction used? You just remember. Now important is Biuret test. Biuret test, when urea is heated at 180 degrees centigrade, the Biuret is formed. Now Biuret mixed with weak copper solution in alkaline medium give purple color if there is presence of protein. So this formula, this principle is used for estimation of protein. Others are denaturation test. These are amino acids which are responsible for giving colors in the different tests. So usually these other tests are utilized for identifying the particular amino acid is there in the solution or not. Biuret test, you read a bit elaborate, you may get short note. Isoelectric pH we have discussed. Denaturation, it is the disorganization of the native protein structure by agents like chemical, it may be, physical, it may be. Denaturation is usually irreversible, but in some cases it may be reversible. Like hemoglobin, when there is salicylate in the hemoglobin solution, the hemoglobin will get denatured. But if the salicylate is removed from the solution, then again the hemoglobin attain its required quaternary conformation, and it gains its function, and it is called reversibility of the hemoglobin due to removal of salicylic acid. So that's why in some cases it is careful denaturation, sometimes reversible called renaturation. If we remove the denaturing agent carefully, then the protein regains its native structure, and that is called renaturation, but it is very rare. Coagulation. Semi-solid viscous, it is produced in coagulation. It is always irreversible, and due to the denaturation it causes coagulation. Example is heat coagulation of the protein. Once heated, it gets coagulated and cannot be brought back to its native structure. Here, basing on the protein's property, some test methods are there to detect protein. These are nephelometry and turbidometry. Nephelometry, when the colloid particles are present in the solution and ultraviolet rays pass, there is scattering of light, and the scattered light is proportional to the concentration of the colloid present in the solution, and wavelength of light used to be 30 to 90 degree. So this principle is used for nephelometry of immunoglobulins. In turbidometry, it is also based on principle of scattering of light by colloidal particles. Only here difference is the degree of emergent light is 180 degree. In case of immunoturbidity, the protein is precipitated by specific antibody, and the turbidity of the antigen-antibody complex is measured. How much antigen is present in the solution, we add antibody. So there is a antigen-antibody reaction, and there is turbidity of the solution. So that turbidity is measured by turbidometry, and the amount of antibody or antigen of interest is measured. Here important things are glutathione, renin-angiotensin system, then your color reactions of out of the color reactions, Biuret test, then properties of amino acid, deamination, transamination, oxidative deamination. These are important things. You must know these things. So this will conclude our chemistry of protein, and next day we will start a new chapter.